In this lecture, we're going to talk about meiosis and sexual reproduction. So meiosis is specifically different than mitosis or asexual reproduction. So let's take a look at asexual reproduction. In asexual reproduction, you're looking at just a single parent, whether that is a single-celled organism that divides via mitosis to generate then two daughter cells, or even in a multicellular parent that produces an offspring via budding. One way or the other, we have a single parent, and the offspring that emerges from that single parent is identical to the parent. The genomes, the DNA, the number of chromosomes are identical. Now, sexual reproduction gives rise to unique offspring. And sexual reproduction relies on the fact that the parents here have multiple sets of chromosomes. Right, so we have a little Y set here and an X set, so an XY here and an XY here. So dad's got two sets of chromosomes, mom has two sets of chromosomes. And sexual reproduction then requires a middleman, a sort of intermediate cell type that's necessary for the production of offspring. What is that? Those are the gametes. So those are the sperm, and those are the eggs. And the sperm and the eggs, unlike the cells from mom and dad here, only have one set of chromosomes. Okay, so dad gives rise to sperm. That is only one set of dad's chromosomes. Mom gives rise to an egg, only one set of her chromosomes. The sperm fertilizes the egg, and now we have the offspring, which again has two sets. And notice now it's a recombination of sets between mom and dad, meaning the offspring is not exactly like dad, nor is the offspring exactly like mom. It's something unique. And it's through sexual reproduction where we get this idea of heredity and variation. Now, heredity means that this offspring, it's got chromosomes that were contributed by dad. It is going to look a little bit like dad, a little bit like mom, a little bit like the brothers and sisters. Like, look at all these puppies, siblings of each other. They all look very similar to each other. However, because it is a new combination of chromosomes, a new combination of genes, it is possible to see variation, to see traits emerge in the offspring that none of the siblings have and none of the parents have. So, for example, this child's very light skin here. So, sexual reproduction gives rise to both heredity and variation. All right, now, with sexual reproduction, we have to come up with some new definitions. And the first is to talk about ploidy, or sets of chromosomes. So what we need to do is we look here at a cell, and let's answer these questions here first. What stage of the cell cycle is this cell in? Well, if we take a look at any given chromosome, and we can answer the second question. We have 1, 2, 3, 4, 5, 6. 6 chromosomes. But notice that each one of these chromosomes is made up of 2 sister chromatids that is connected by a centromere. Okay, so they're duplicated chromosomes. Okay, but there's still a single chromosome here because they are connected by a centromere. So the cell cycle that they must be in is probably pretty darn close to metaphase, although they're not lined up there. So they're somewhere in the mitotic phase, definitely beyond the S phase of interphase because they are duplicated chromosomes. And probably in somewhere in mitosis because they are fairly condensed. Okay, now, this equation here is going to be important. Okay, this is going to be the equation that tells us the ploidy of the cell. Now, after the equal sign, That number there, 6, tells us the total number of chromosomes in the cell. So let's count them. 1, 2, 3, 4, 5, 6. So that's why there's 6 there. N means sets. How many sets of chromosomes are there? This is the tricky part. What we have to do is look at a single chromosome and see if there are any other chromosomes that are about the same size and about the same shape as this chromosome here. And we count how many there are like that. Well, if we look, no, yes. So these 2 chromosomes are about the same shape. The centromere is about in the same location. They're the same size. And so we call these 2 chromosomes, they are homologous chromosomes. Okay? These 2 here, they're labeled here. These 2 are homologous chromosomes. So what we have here is 2 sets of chromosomes. We have the set of chromosomes in red, the little guy, the medium guy, the big guy, and then we have their homologous set. This blue little guy, this blue medium guy, and this blue large guy. Okay? So we have 2 sets of chromosomes. So what we say is this cell is 2n equals 6. 2n means we have 2 sets of chromosomes. All right. So let's talk a little bit more about homologous chromosomes. Homologous chromosomes are completely different than sister chromatids. These are 2 separate chromosomes. Each one has been duplicated, so each one consists of 2 sister chromatids connected by a centromere. Now, what makes these 2 chromosomes homologous? Well, as I said before, they are about the same size. They have a centromere in about the same location. If we were to stain them with particular stains, they would stain in similar ways. Most importantly, they have the same types of genes in the same locations. That means they have the same loci. So for example, if this blue homolog has the eye color gene here, then the eye color gene is going to be in the same place on its homologous chromosome. Now, I mentioned that they're similar but different. Well, what do we mean by different? Well, what I mean by that is even though they both have the same, they have the eye color gene, so the same type of gene in the same location, the exact version of that eye color gene can be different. So here on mom's chromosome, it's the blue-eyed allele. Here on the other homologous chromosome, maybe from dad, it's the brown eye color allele. Okay? So that's what I mean by homologous chromosomes are similar but different. They have the same types of genes in the same locations, but the alleles of those genes might be different. Now, let's look at something called a karyotype. Now, a karyotype is an organized array of somebody's chromosomes. One of the most important uses of karyotypes is to look for broad chromosomal abnormalities in a developing fetus. So let's say a woman is pregnant, and right around 12 weeks or so, what a doctor can do is go in and take a sample of the chorionic villi. Now, the chorionic villi is not part of the fetus itself, but its cells share all the same DNA and all the same chromosomes as the fetus. And so you can take a biopsy of these chorionic villi cells and not damage the fetus at all. So the doctor takes out those cells. The doctor then gets those cells to divide and divide and divide, and then adds a drug called colchicine. And that arrests all of the cells at metaphase of mitosis. And that's important because remember, metaphase of mitosis is the phase at which chromosomes are the most condensed and easiest to see. The doctors then break open those cells and they digitally arrange the chromosomes in order from chromosome 1 to the 2 sex chromosomes here. Now, humans have a total of 23 chromosomes, or 46 chromosomes in 2 sets. So, this is chromosome 1. Now, each one of these, I know you can't see it, it's really difficult, but each one of these that you see is made up of 2 sister chromatids. So, 2 sister chromatids here and 2 sister chromatids here. The resolution is such where it's difficult to see the independent sister chromatids. Which means one of these is chromosome 1 from mom, one of these is chromosome 1 from dad. They are homologous to each other. So humans have 23 pairs of chromosomes. And as you can see, the biggest chromosomes are the 1, 2, 3, 4. The smallest chromosomes are 20, 21, 22. And then we have these 2 chromosomes here. These are called the sex chromosomes. You can either have an X or a Y chromosome. So either 2 X's or an X and a Y. And that will determine whether you're male or female. So let's answer these questions. Is this from a diploid or a monoploid cell? Well, we've got 2 sets here. For every chromosome we see, we've got a homolog. So it must be diploid. If it was monoploid, we would look at chromosome 1 here and we would not find another chromosome 1, just 1. Is this person male or female? Well, look at that, right there, 2 X chromosomes. That's a little daughter. So what does 2n equal? Remember, 2n equals, after that equal sign, it's always the number of chromosomes. Well, we have 23 pairs, so 23 times 2 is 46 chromosomes. What does n equal? N is the number of chromosomes in a single set. Therefore, if 2n equals 46, n equals 23. How many sets do we have? Just 2 sets. The set we got from mom and the set we got from dad. How many autosomes are there? There is 44 autosomes. Autosomes are all of the chromosomes other than the sex chromosomes. And finally, this is one of the most important reasons why someone would do a karyotype on a fetus, so on a very early during pregnancy, is Down syndrome begins to occur more often when women are over the age of 35 and pregnant. And Down syndrome is also called trisomy 21. What that means is chromosome 21 is represented 3 times. So let's take a look. Here's chromosome 21. How many chromosome 21s are there? There's 1 here, good, from mom, 1 here, good, from dad. Is there a 3rd one? Nope. So we're good to go. This child does not have Down syndrome. And in fact, what you guys are looking here is my daughter's first baby picture. This is my daughter's chromosomes, which means each one of these chromosomes, one of them is mine and one of them is my wife's. So I guess it's kind of weird that you guys are looking at my chromosomes. Anyway, yep, this is my daughter's karyotype. So let's take a look. Now that we understand what a homologous chromosome is and what ploidy is, why do we need meiosis? Well, here's the problem. If sperm and egg if sperm and egg were just created via mitosis, they would have the exact same number of chromosomes that dad had and that mom in mom cells. So 2n equals 4, 2n equals 4. Which means if that sperm fertilizes that egg, suddenly now our offspring has a 4n equals 8. We've doubled the number of chromosomes in that offspring. And then that offspring has more offspring, that chromosome will double. So that means that in the process of gametogenesis, in the process of producing an egg or producing sperm, that's called gametogenesis because sperm is a gamete, egg is a gamete. There has to be something special, something called reductive cell division. And that's what meiosis is. Meiosis is a way of taking the diploid cells of mom and dad and creating sperm or eggs that have half the number of chromosomes, that have only one of the two sets. And so that sperm can then fertilize the egg during fertilization, and that restores the diploid number in the embryo, which will then become an adult. And within the ovaries or the testes, meiosis will occur, generating sperm or eggs, and that process will repeat over and over and over again. So let's practice this. Why don't you pause this and take a moment to try to answer these questions regarding these chromosomes. All right, let's do it. How many chromosomes are there? Well, there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 chromosomes. Okay, so that is going to be that number after the equal sign. How many sets of chromosomes are there? Well, we've got a red A, B, C, D, and then we have a blue A, B, C, D chromosome, and we have a black A, B, C, D. We have 3 sets of chromosomes. Okay? So what that means is we have a situation where 3n equals 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 3n equals 12. What is the ploidy? Well, the ploidy is that 3. How do we figure out the ploidy? Well, you look at a single chromosome, B, and you look at how many chromosomes look like this one. We have 1 itself, 2, and 3. Do we have, I think that's it, right? A red, a blue, and a black. So the ploidy is triploid. Each chromosome has a total of 3. So 3n equals 12, and therefore n equals 4. There's 4 chromosomes per set. An A, a B, a C, and a D. All right, so let's take a look at the overview of meiosis. If we are looking at a cell, before the S phase of interphase, what we see is that each chromosome, we have this chromosome here, and we have this chromosome here. They are homologous to each other, and they don't dissociate or anything. We know that they've got the same loci and stuff, but they don't hook up at all. They don't interact with each other at all. But eventually, we're going to go through the S phase of interphase. So each of them are going to duplicate. So now we have this chromosome is made up of sister chromatids. We've got its homologous chromosome made up of sister chromatids. And then what happens is during meiosis, the first part of meiosis, the 2 homologous chromosomes separate. So this is immediately different than what occurs in mitosis. In mitosis, all that happens is the 2 sister chromatids separate. In meiosis 1, the 2 homologous chromosomes separate. We don't get any breaking of centromeres. And then in mitosis, so now we have a situation where this has this chromosome, so we've already halved the number of chromosomes, but the problem is each chromosome that's in here is duplicated, so we have to then separate the sister chromatids, and that's the job of meiosis 2. So what we end up with is we start with 1 cell, and we end up with 1, 2, 3, 4 cells. The 1 cell we started with had 2 chromosomes in it, and the 4 that we end up with only have 1 chromosome in each one. So in the next video, we're going to look at the details of meiosis 1 and meiosis 2.